Good afternoon, friends. In this session, we will be covering the second block of the course 7, Food Testing and Evaluation. We have Dr. Saxena with us, who is subject expert for this. Friends, we have already covered in the first session the important aspects of the food quality. In this part, we will be covering the tools. What are the different tools for testing and evaluation of the food quality parameters? Like, what are the physical methods, what are the chemical methods, what are microbiological methods, and what are the sensory evolution methods. So these aspects will be covered in this session. Friends, as you know, the special feature of this program, diploma program in value added products from fruits and vegetables, is to develop entrepreneurs in different dimensions of the food processing or fruit and vegetable processing. As you have heard in the first session, the quality control is becoming an important aspect, and many of the entrepreneurs can go and set up their own laboratories. They can be manager or can set up their own small quality control labs. So what type of a quality control lab looks, particularly for processed food products? So we will see a program, and then Dr. Saxena will be telling you about the different methods. So let us see a program, testing and evaluation of processed products on this, and then we will come to the main discussion on testing and evaluation. This
Hello friends, to check the quality, safety and adulterants of the food products, you need a food testing laboratory. And this is the duty of the entrepreneurs to ensure that food testing laboratory is equipped and accredited. Equipped means that laboratory has sufficient equipment to test the required parameters, and it is accredited. Accreditation means the third party insurance that the laboratory is technical competence. To ensure the technical competence, in our country we have a board that is known as NABL. It is called National Accreditation Board for Testing and Calibration Laboratories. This board belongs to Ministry of Science and Technology, and they accredit the laboratory on the basis of ISO 17025-2005 standard, which is a global standards. Anywhere in the world, the laboratories are accredited and their technical competence are ensured on the basis of ISO 17025. To see the food testing laboratory, there are some regulations which has to be followed. Like in the food testing laboratory, we have to ensure that there is no outside contamination. That's why you can see that we have a double door entry to check any outside contamination within the lab. The second very important point is that the person who is coming with the samples is not directly interacted with the technical people working in the laboratory. So these people are directly directed to the sample cell, where the person responsible receives the samples and ensures that he has taken care of all the testing requirement of the customer. The second important point within the sample cell is that the person sitting there, he put the code number on the sample. So the person who is testing the particular sample doesn't know the samples belongs to whom. So that's why the integrity and the confidentiality is maintained. And generally, if you see in the food testing laboratories, there are 3 sections, like instrumentation, where the different instruments are there, the chemical, where the wet chemical analysis is being done, the microbiology, where all the pathogens are being tested. Now I'll let you know each and individual equipment and their usage in the laboratories and the different sections. Now we are in the microbiological section, and this is the inoculation room, and it is nothing like, it is like a operation theater. We have to ensure that there is no outside contamination, and our colleague is working on the laminar flow. This equipment is the laminar flow, where we ensure that the working platform is sterile. So the air which is coming is zero filtered, so there is no microbe on there, and the samples which are being inoculated there, the contamination which will come out only from the sample, not from the environment. As you are aware, the microbes cannot be seen with the naked eye, so these samples are inoculated into the different medias, and after inoculating of these different medias, they are being incubated at different temperatures. So these are the incubators. Here you can see, once a particular temperature and time is maintained, these bacteria grow in the colonies, and here we can identify and we can see that. It is also important, while analyzing the different bacteria, the different temperature is required. So we have a different incubators for different temperatures, the 40 degree, the 37 degree, and so on. Now you see, this is the colony counter. Under this colony counter we put the plate where the pathogens are there, and we can count the pathogens. We can identify the species by putting the standard slide under this microscope after proper magnification. And now we can move to the next section. Now we are in chemical section. I will let you know about the utility of some of the equipment in chemical section. This is fat extraction unit, and by this unit we extract the fat in any food product and know the quantity of the fat in a sample. Like this is a biscuit sample. We would like to know how much fat is in this biscuit. What we have to do, we have to crush the sample and make into the powder form, and this powder form sample we will put into this thimble, this white color unit, and we put into the soxlet. This apparatus is known as soxlet. This is the flask, soxlet, and condenser. So we put the sample in this white color this thimble, and ether is put into this flask, and condenser is put over this soxlet extractor. Then we set the certain temperature. This water is circulated through the condenser, so ether is cooled. So, and this ether is circulated again and again. After certain hour of extraction, like you see, the ether is going down, and the fat is dissolved into the ether, and ether along with the fat is collected into the flask. The flask is taken out, and the ether is evaporated, and the fat content is weighed into the analytical balance, and we know how much percentage of the fat is there in the biscuit. Now I'll inform about another equipment. Now I will inform you about the protein estimation. If you see any food product, on the label the nutritional facts are there. which contains protein, fat, carbohydrate, and how much energy a product is getting. To make sure to estimate that how much accurate these are, we have to estimate protein, fat, and carbohydrate. And this equipment is used for the protein estimation. For example, this is the juice sample, fruit juice. We have to take into this flask and digest it. The digestion is being taken at a particular temperature, and then it is to be heated at a particular temperature, and the sample is digested and condensed. Once the sample is digested, it becomes very clear. Once the sample is clear, then you take for the distillation and titration. By this way, we can estimate the protein in any sample. This is the equipment known as profound color grader. By this equipment, we can know the color of the honey, and this is the international standard requirement. What we have to do, there is a cuvette. We have to put the honey into the cuvette, and we have to put on the light and match the color. And after matching the color, we can note down the color of the honey, how much color is there, and it is recorded in mm. Now we are in the instrumentation section of this laboratory. You see there are different equipments, and I will explain few of them. This is analytical balance, and this balance is basically the backbone of the laboratory. You start any analysis, you have to use this analytical balance, because you have to weigh accurately the chemicals, the reagents, and the sample. And this is a very sophisticated balance where we can weigh up to 0.01 milligram of any chemical or any sample. Then I move to the next equipment. This is spectrophotometer, where we do the estimations of the different chemicals like iron, phosphorus, or the color. So these are the different colored samples, or the color can be developed by a chemical reaction, and this can be estimated by this equipment, and ultimately the concentration can be known. For example, if you want to get the iron in a particular sample, you develop the color, and then get estimated by this equipment. This is water activity meter, where we can get the water activity of a product. Say this is the moong dal sample, we have to crush it, we have to put into the cuvette, and then we put into this sample cell, and after that we put this hygrometer, and we can read the water activity. This indicates the probability of having the presence of the pathogens in a particular sample. Now this is the pH meter, where we estimate the pH of any juice or any water sample, any liquid sample, we can estimate the pH. First we have to calibrate this equipment by the standard pH 4 and 7. We take the sample into the flask, and then we get the pH, and the directly digital reading comes onto the equipment, and we can note the reading. If the pH is below 7, it indicates it is acidic. If pH is more than 7, it indicates it is basic in nature or alkaline in nature. This equipment is the conductivity meter, and conductivity meter is generally conductivity is required to estimate in the water sample how much conductivity is there. So we take water sample into the flask, directly put the electrode into it, and conductivity can be read on this equipment. You have seen a food testing laboratory which is accredited by NABL based on ISO 17025. It ensures its technical competence. Technical competence in the sense that the manpower is qualified, equipments are calibrated and up to the mark, and the system is being followed. You have also seen all the three sections of the laboratories, like microbiological section where you have seen the microbiological testing. In the chemical section you see all the chemical analysis. In the instrumentation section you have seen that there are different equipments, including sample cell where the samples are being registered and integrity is maintained. You have seen most of the common equipments which are lying in the laboratory, and they are used for the food testing as per the national and international standards. Thank you.
So friends, now I will request Dr. Saxena to cover the other important features of the block. Dr. Saxena, please.
Thank you, Dr. Salooja. We have seen in the laboratory different equipments and how these equipments are being used to analyze the physical, chemical and microbiological parameters of the different food products. And now we will discuss in detail what are the principles to analyze different parameters by using these equipments. If you see the analysis of any food product, if you categorize the analysis of different food products, they are mainly in physical, chemical and microbiological. One of the important parameter is also the sensory evolution, which is the physical parameter. This comes under the physical parameters. Now if you see any food product or going to buy any food product, our sense organs, we call as indriyas, which play a very important role. If you are going to buy a product into the market, the color is very, very important aspect, and this is the physical color. And the color is already perceived in our mind. If suppose we are going to buy a laddu, it is already in our mind that laddu should be of kesari color. If laddu is white color, nobody is going to buy this as laddu. Similarly, like we are going to buy a vegetable, it is a bhindi. Bhindi means the green colors. So the color is very, very important. What we eat, it is first, until unless it is attracted to our eyes, we are not going to eat it or buy it. So the color is very, very important aspect, an important parameter. Color play a very important role in the assessment of the quality of the food products. Like tomato ketchup, if it is a red, then it's okay. If it is going to be brownish, then something wrong. So while seeing the product, while seeing the color of the product, the consumer perceive the quality of the product. So the color is a very, very important parameters, and there are different equipment, there are different methods by which color are being measured. Color can be measured by matching. You have a standard color and you can match your product is of that mark. Color comparing, you can compare the color of the product with your standard. Quantitative color measurement, you can do by spectrophotometer, you can do by Hunter scale, you can do by tintometer. So different food product colors are, different equipments are being used, and there are different principles are there by which the colors are being measured. Now if you see the color matching, so there is a color standard scales are available by which the product is being put and color is being measured. The quantitative color is being measured by the spectrophotometer. You have seen in the laboratory, and I have brought this equipment here also on the slide. and it is basically based, color measurement is based on the Beer-Lambert law. What is Beer-Lambert law? If a spectrum of light passes through a medium, either it is absorbed or it is transmitted. So the color is being basically measured either in terms of transmittance or in terms of absorbance. And if the transmittance is high, the color is going to be low. Similarly, absorbance is high, then transmittance is going to be low. This is transmittance versus the absorbance is inversely proportional. So you can measure the color of a product by spectrophotometer at a particular wavelength, and that is the visible range where you can visible and you can see or you can measure the absorbance. In the spectrophotometers, what happens? Once you put a sample in the cuvette and pass through a light, according to the color of the sample, it has a absorbance at a particular wavelength. So you have to set the wavelength of the equipment and then you can measure the color. Similarly, there are other methods of measuring the color like tintometer. What happens in tintometer? There is a standard color plates are available and there are circles by which the standard colors are there. Like you are going to measure the color of a beer sample. So you put the beer sample in the cuvette and put into the tintometer cell and you rotate the standard, and once standard is matched with the beer sample, then you can say it is matches with this LBU color and you note it. Like in the previous session you have seen, for the measurement of honey, the color measurement of honey, there is a profound scale. So there is a standard scale, you put the honey in the cuvette and you match it. Similarly, in tomato products, there are the Hunter scale. Hunter is a scientist who developed the Hunter measurement system. So it is basically on the basis of reflectance. So the sample is put into the cuvette cell and then the reflectance is measured either on the A or B, and ultimately A reflects for the redness and B reflect to the brownness or yellowness, and ultimately the LAB ratio is a Hunter value. So if AB ratio is more than 2, the product is going to be more reddish in color. So like tomato and tomato product or tomato ketchup or tomato paste, the color is being measured with the Hunter scale. So we have to summarize the color in the three major equipments like tintometer, Hunter scale or spectrophotometer. These are the three equipments which are being majorly used for the measuring the color. And color is higher or lower doesn't indicate the quality. It is basically the perceived, conceived ideas. Okay, what should be the color? Even then, suppose the color is high, like beer color is more dark, it doesn't mean that it is a good product. Or the color is very low, it doesn't mean the quality is okay. So at a particular color, as a perceived, conceived idea, what should be the color of a good product and which has to be measured. And accordingly, the national and international standards are formulated. Like for example, the water. Water, if you see the color, it should be colorless. But ideally, quantitatively, the color cannot be 0. If you see the IS standard for water, IS 10500 or IS 14543 for packaged drinking water, the color should be maximum 5 Hazen unit. So this color can be measured by spectrophotometer. By naked eye you cannot compare whether it is having some color or not. Once there is a color, then you can say that it is a color or pale color, but it should be as per the standard. To summarize the color doesn't indicate, as such color doesn't indicate the quality is good or bad, but definitely a definite color is required for a quality product. Like tomato paste, it should be red. Red means in what terms? Like Hunter value, it should be above 2. If above 2 means LAB ratio is more than 2, the product is very good. Now come to the next parameter, the physical parameter, that is viscosity. Viscosity and consistency. And these are being measured by different type of again equipment. It is again a physical parameter which basically measures the flow behavior of the fluid. There are Brookfield viscometer, the Bostwick consistometer, Oswald viscometer. There are different type of viscometers are there. And viscosity basically is measures the flow behavior of the liquid samples. And there are two type of liquids, Newtonian liquids and non-Newtonian liquids, which follow the Newton law and which do not follow the Newton law. If you see in this picture, there is a viscometer, and we are measuring the viscosity of a liquid. It's a gum, liquid gum sample, and these are the spindles, different type of the spindles. These viscometers can measure the viscosity of a product from 100 CPS to 1 million CPS. So there is a different wide range of the viscosity we can measure by this equipment. And you see here in this cabinet there are the different spindles, 1, 2, 3, 4, 5, 6, 7, and spindle can be changed from this nut. And according to the presumption of the viscosity of a product, we can put the spindle of a size, okay, and then we put the speed, we can set the speed, and then also can be set the temperature, and viscosity can be measured. So from low viscosity to higher viscosity product can be measured here. And again, viscosity is a physical parameter. So viscosity high or low doesn't indicate the quality, but for a particular product a required viscosity is required, so that product should have a required viscosity. Now the second equipment is Bostwick consistometer. By this we measure the consistency of the product. Like suppose a product having a 2 layers of the product. A viscosity means the product having a, suppose a tomato paste. So it has a 1 layer and it is a down layer. It is a layer of the particles. So one layer try to flow down upward, and another layer, because of the gravity, it put the pressure to the down. So it resists to flow the product. So how much this product will flow on this consistometer can be determined, and this can be determined as per the international standard within 30 seconds. So there is a channel and there is a cuvette. We put the product in the channel, in the cuvette, and there is a trigger. We fill the product and then allow to flow this product up to 30 seconds, and then we measure how much cm it flow, and that is the international standard. Similarly, the Oswald viscometer, there is a 2 tubes, glass tubes, and 2 bulbs is there, and you suck the product into one tube and start the stopwatch and to see how much it passes through the valve, and it indicates the physical parameter and the viscosity. So different type of liquids, the viscosity and consistency can be measured by Brookfield viscometer, by Bostwick consistometer, by Oswald viscometer. And again, as per the national and international standards, we have to use the different equipment for the measurement of the viscosity. I repeat again, viscosity as such, low or higher, doesn't indicate the quality of product, but a definite viscosity, a range of viscosity is required to measure the quality of product. Like gum should have a very high viscosity, the milk juice should be having a low viscosity, or the milk product should have a low viscosity, something like that. So we can measure the viscosity and consistency of the products. Another physical property is the texture. It is a property of the food which is associated with the sense of feel or touch experienced by finger or mouth. Textures, how you can measure the texture of a product? Either you touch it, or once you put into the mouth or you eat it, you feel that how the product is.
or sometimes physically you can see how the texture of the product is. It can be quantitatively analyzed by texture analyzer by applying the different forces through different devices on the food. Like suppose, if you see the apple, from outside apple is seen very clean, red in color. But until unless you touch it, you will not feel that it is rotten from inside or it is soft from inside. And this is the physical parameter to judge the texture. But there is now equipments are available by which you can measure the texture. You put a device on the product and then you apply the force. By applying the force, when the product is ruptured, that is basically determine the texture of the product. Similarly, the bread, the biscuit, then other things. Like physically how we determine the texture of biscuit, the crispness or the papad, we just try to make into two pieces and then we'll see how crisp this product is, and that basically indicate the texture. If suppose the papad is soggy, it absorbs the moisture. If you try to break into two pieces, it will not break it. Suppose in the biscuit the moisture is very high, it will not indicate a good texture of the product. So similarly, physically by the qualitative method and quantitative method, we can measure the texture of the different products. Then we come to chemical analysis of the different food. This we have seen in the last episode, that this equipment is the protein analyzer. And if you do the protein analysis of a product, it has basically three steps. One is digestion, another is distillation, and third is titration. And protein is one of the important constituent of the food, which is required for the body tissue building. So every product has some of the amount of the protein. And protein is basically analyzed once you take out the nitrogen and then you calculate on the basis of the particular factor. So here we see the product is being crushed into the powder, put into the flask, put the hydrogen peroxide and heat up to the 450 degrees centigrade, and sample is digested, and all the hazardous organic matter through this condenser and passes through the drain. And once this sample is digested, it is clear, it has nitrogen content in it. Then this nitrogen is being distilled by the distillation apparatus, and then it is being titrated total as a nitrogen. And once you come with the nitrogen and then you apply the factor, in the certain product it is 6.25, in milk and milk product it is 5.75. For different product different factors is applied, and then you calculate the protein content in a food product. Another major constituent of a food is the fat. Fat you can feel, but you cannot quantify until unless you take out from the food. And how it can be taken out from the food? Because fat is soluble in the petroleum or diethyl ether. So you have to mix the food with the diethyl ether or petroleum ether, and then you take out. So this is the solvent extraction, Soxhlet extraction units are there, you see in the figure, and you put the sample into the thimble, put into the Soxhlet, put the petroleum ether into the flask, and it is a low boiling organic solvent, so it boils at 60 degrees. So you maintain the temperature 60 degree, and through the Soxhlet condenser it passes again and again and again and again, and depending on the nature of food, it takes 5 hours, 6 hours, 8 hours, sometimes it is 18 hours. And then once the fat is soluble completely into the diethyl ether and come into the flask, and you take out. And once you take out and take the weight of the flask, and it can be calculated, the percent fat is equal to weight of the ether soluble material divided by the weight of sample how much you have taken into 100, which indicates the percent fat into the food product. The another major constituent of the food is the carbohydrate. The estimation of carbohydrates can be done by two methods. One simple method is, if you know the moisture, ash and protein and fat, you directly minus into the 100, then you will get the total carbohydrate content. Otherwise you can apply by the sugar because carbohydrate is nothing, it is sugar. So you can apply the analysis method for the sugars and you can get the percent total carbohydrate. But ideally you can also get it once you have a moisture, S and protein and fat, you make it total and minus is 100 is the percent carbohydrate. And you take any food, out of that, why we take the food? Food gives us energy, and energy is being used for our different functions. If we don't take the food, what will happen? We will not get the energy and we will not be able to function. So how much energy is being taken by our body from a food? How it being calculated? It is being calculated based on the protein, fat, carbohydrate, on the basis of that. So 1 gram of fat give you 4 calorie energy, 1 gram of carbohydrate, it gives you 4 calorie of the energy, and the fat gives you 9 calorie. So more than the double. If fat is more, you will get more energy, you will get more calories. So the total calorie of the food is percent protein into 4 plus percent carbohydrate into 4 and plus percent fat into the 9. It is a total is the total calorie you are getting per gram of food. So nowadays, recently the government of India has put a notification that every packed food product should have the nutrition value on the label. So it basically called as nutritional fact. So tomorrow if you are going to buy any product into the market, you will see after consuming of 100 gram of this food, how much energy you are going to get it. Whether it is a milk product, whether it is a biscuit, whether it is a chips, or whether any type of packed food product, it is a revolution now of the government of India, which is going to be implemented by February 2008, and every food product will have this information, so consumer will be aware that how much energy he is getting it. And how the processor they will get it? They will get it, their product tested in the laboratory on the basis of protein, fat and carbohydrate. They can calculate it and they can put into their product for the consumer information. If you go abroad, any food product, if you see, it will have a nutrition fact on the label, and people will be, according to their choice, their health consciousness, they can take their product. Now the second category is a microbiological examination of the food and water. Generally, for the hygienic point of view, there are 3 parameters: the total plate count, the total coliform count and total fecal streptococci count. They are being is a hygiene indicator of either food or water or any utensils. And the different steps for the microbiological analysis we have seen in the laboratory, but the major part is the media preparation, inoculation of the sample, incubation, and ultimately we can get it total colony count. Media preparation, because as I mentioned that these microorganisms cannot be seen by naked eye, so we have to give them food to grow into colony. Colony means the number of the units, and that is called as colony forming unit, CFU. And then you can, after particular incubation, you can find out how many colonies are there. Microbiological examination of the packed food. Basically there are, if you have a packed food, you can also determine on the basis of microorganisms how the food is spoiled. Okay. So that food product, particularly the packed food product, can or Tetra Pak, they are being incubated at 37 degrees centigrade and 55 degrees centigrade, and after that they do the physical examination. Once you incubate at 37 degree and 55 degrees centigrade, the bacteria present into the food product, they multiply and they grow because food is already available into the food product. So at this, once the temperature is given, they multiply, and once they multiply, they produce the gas, and once the gas is there, then the product is going to be bulged, the package is going to be bulging. So that is one of the method of examination. If your can is being incubated for 37 degree or 55 degree centigrade, if there is no bulging, it means product is okay. If product is bulging, it means there is a contamination and product is going to be spoiled. And another examination, you can cut the can and drain your product, and after seeing the inside the can, you can find out there is some problem and there is a spoilage of the food product. Then there is a direct microscopic examination. You can take the food and see under the microscope, under the magnification, that your product is going to be spoiled. Then there are different other aspect of the food product is the sensory analysis of food. Sensory evaluation, it covers the sight, smell and taste. How it looks, how it smells and how it tastes. And it can be done qualitative and quantitative, both type of tests are there. And they are affective test, acceptance test, preference test and hedonic test. And the latest development is there because these are all on the basis of the human beings, perceptual human being test. But there is now recently development that there is a electronic equipment we call as electronic nose. So the total flavor profile of the food product can be analyzed. And we can also measure the consistency of the different food in that case. Affective test, acceptance test, preference test, hedonic test, these are the different category of sensory evaluation. How the sensory evaluation is done in the lab? Because there is a panel of 5-6 people who taste the same food product without seeing each other, So there are different cabinets are there, and then these panelists are sit, and one examiner or one the leader, they give the coded samples to these panelists to identify how this product they are rating it. Effectiveness, whether they give the score, whether the product is acceptable or not. Similarly, preference test, they are given one product and asked that whether it is matching the requirement of the another product or not. So like you develop a cola product and you want to see the match with the Coca-Cola, then you given a Coca-Cola as a coded product and you will ask the panelist that you match this product, how it tastes, how it smells and how it taste and flavor is like that. So the panelists do this job, and on the basis of their scores, the product quality is being monitored or being defined. Similarly, the major precautions has to be taken while testing the panelist. They should not see each other, because this is the psychological. If I am testing the product this water, you are also testing this product this water. If I take this water and by face feeling, if I do not find the good taste, you can also recognize that this product is not good. So that will be the bias. So while testing the sensory evaluation, you should make sure that there is no biasness is there. Thank you.
Thank you, Dr. Saxena, for very comprehensively and with examples covering the very complex subject, testing and evaluation. Dr. Saxena covered the physical test, that is color, viscosity and texture. Then in chemical test, he covered the protein, fat and carbohydrates. And in microbiological, the plating, and then the coliform count, and then the direct examination under this. And then he also covered the sensory evolution part of this, that how do we do the sensory evolution, and he also touched the new electronic nose and how the panels should not be biased. So with this we cover the second block of this course, testing and evaluation. Friends, if the questions are there, please welcome. Thank you very much for listening to this. Thank you.